virtual class in which we are continuing the topic molecular basis of inheritance. Today we are going to learn one very interesting concept called regulation of gene expression. Gene expression is resulting in the formation of polypeptide chain that gives particular type of protein which is responsible for the metabolic activities of the cell. In case of eukaryotic cells, the expression of gene is going to be controlled in 4 levels. They are the first one called transcriptional level, in which the synthesis of mRNA takes place. Then processing level, in which the mRNA will be processed, called splicing, in which all the introns will be removed and exons will be joined to form a functional mRNA. Later, the third step, in which transfer of mRNA to cytoplasm, which is going for translation. Then the last one is translational level, in which the process of protein synthesis takes place, that is called translation, as we have discussed in the previous class about translation. So these are the 4 different levels which is going to work in case of eukaryotic cell. So these are the 4 levels which are going to work in case of eukaryotic cell, resulting the gene will going to form an polypeptide chain. Similarly, in prokaryotes like P.U.College or number of other bacteria, the process of gene expression is controlled by operon. Operon is nothing but an unit which is made up of set of genes like structural gene and control gene. So in other words, operon is a cluster of genes in which more than one form of gene will be clubbed and it will work together, called as an operon. So in prokaryotes, there is a regulated gene called operon, which is made up of structural gene and control gene. There are 2 sets of genes which will need to work together to form an operon. So to explain the function or regulation of the gene, there was a famous set of scientists by name Jacob and Monod. Jacob and Monod, in the year 1961, they proposed one concept of regulation of gene called lac operon concept. This lac operon concept was one of the famous model which has been put forward by Jacob and Monod in the year 1961 to show how the gene will work in case of prokaryotic cells. As we know, prokaryotes are having the operon. Here the lac operon says lac indicates lactose sugar, and operon is the regulated gene or the bundle of structural and control gene. So in the presence of lactose sugar, the lac indicating lactose sugar, which acting as an inducer which makes the gene to get activated. So in the presence of lactose sugar, gene will work. In the absence of lactose sugar, gene will fails to work. Or in other words, in the presence of lactose sugar, gene will be switched on to produce number of enzymes. And in the absence of lactose sugar, that is inducer, the gene will fail to produce any enzymes, and that is what we call switch off condition. So this has been very clearly explained by Jacob and Monod with a concept called Lacoperon concept. This concept they are explained by taking a model of E. coli bacteria. In E. coli bacteria, which is one of the examples for prokaryote, there was a cluster of gene, what is called as an operon. Here, the lac stands for lactose sugar and operon is a cluster of genes, what we call as a regulated gene, in which the structural and control gene will work together in the form of one single unit. Operon is made up of control gene and structural gene. That's why the name has been given called lac-operon. Lac for lactose, operon is cluster of gene. So there are two sets of genes which will going to work here. One is the structural gene in the form of lac-Z gene, lac-Y gene, lac-A gene, which is responsible for the production of three different enzymes. That is, lac-Z gene will produce beta galactosidase enzyme. Lac-Y gene will produce permease enzyme. Lac-A gene will produce trans acetylase enzyme. So these are the three sets of enzymes produced by structural genes in E. coli or in a Lacoperon concept. To produce these three genes, it is the control gene which is going to regulate or which will control the function of structural gene by three sets of genes, that is promoter, operator and I gene or inducer gene, which is responsible for the production of a protein called repressor protein gene. So these three set of genes called control gene, promoter, operator and repressor gene, these three will going to work for the production of three enzymes which is regulated by the control gene. I'll repeat, the control gene regulating the function of structural gene for the production of three enzymes, that is beta galactosidase, permease and transacetylase. So these three enzymes will be produced in the bacterial cell only when there is a presence of lactose sugar. In the presence of lactose sugar, gene will be active or what we call as a switch on condition. In the absence of lactose sugar, it will be inactive or it will be switch off condition. That we'll see in the next diagram. Well, lactose sugar is acting as an inducer which makes the gene to work. In the presence of lactose sugar, which act as an inducer, gene will be active or what we call switch on condition. In the absence of lactose sugar, gene will remain inactive condition or what we call switch off condition. So when it will be switch on and switch off and what are the things will going to happen, we'll see here. So in the presence of lactose sugar, to keep gene switch on, it is the Ig which is responsible for the production of repressor RNA. It is called repressor mRNA. This will going to involve in the production of a protein called repressor protein. It is called repressor protein. So in the presence of lactose sugar, in the presence of lactose, which is acting as an inducer, the repressor protein will get inactivated. It will undergo inactivation in the presence of lactose sugar, resulting the repressor protein will bind with inducer and it will form inactive repressor protein. As it becomes inactive, it fails to attach to the operator region, resulting all the three genes, structural genes, that is Z, Y and A, which is called as a structural gene, which is responsible for the production of three different types of RNA. So here, transcription takes place, resulting it will form an RNA. So that transcriptional mRNA undergoing a process of translation. So as we know, translation is a process in which the process of protein synthesis takes place. That is what we call polymerization of amino acids takes place. So here, resulting each one of this mRNA will undergo polymerization, resulting it will form three different types of enzymes. That is beta galactosidase and here permease, and lastly the lactic A gene will produce transacetylase. All these three enzymes will be going to synthesize only in the presence of lactose sugar. So this is what we call gene switch on condition. What will going to happen if there is no lactose sugar? It is called switch off condition. That we'll see here. In the absence of lactose sugar, as usual, this gene will produce mRNA called repressor mRNA. This repressor mRNA will going to produce the protein called repressor protein. This repressor protein goes and attaches to operator. As it will attach or bind to the operator, the structural gene fails to work. The three genes fails to work. So as a result, no transcription. That means no mRNA is going to synthesize. So in the absence of mRNA, no protein synthesis takes place. So no transcription, no enzymes. So enzymes also will fail to produce. So this makes the gene to remain inactive condition, are also called switch off condition. So in the presence of lactose sugar, the lactose sugar makes this repressor protein inactive, resulting gene will work for the production of three enzymes, that is beta-galactosidase, permease and transacetylase, whereas in the absence of lactose sugar, the repressor protein goes and binds to the operator region, resulting blocking all the three genes, which is not able to undergo transcription, resulting no mRNA and no enzymes will produce. That is what we call switch off condition of the operon. So I'll conclude here. In the presence of lactose sugar, which acts as an inducer, repressor fails to bind to the operator, makes the gene to work, enzymes will produce, and that is what we call switch on condition of the gene. In the absence of lactose sugar, repressor protein remain active and it will attaches to the operator gene and fails to produce any RNA from the structural gene, resulting no enzymes, and it is called switch off condition. So this is what we call the lac-operon concept, one of the important concepts asking for 5 marks in the board examination. Thank you.